So let me begin again. I'll share the screen. Right. So I was talking about the supporting medium. Okay. The supporting medium, since this supporting medium or the paper itself is not involved in any kind of interaction with the sample components, we expect the supporting medium to be very inert. Okay, inert. What is the meaning of inert? It should not itself react or interact with the sample components. It should only serve the function of a support medium for the liquid or water, which is the actual stationary phase. Okay, and another thing is it should be stable. It should not get destroyed or damaged very easily during the chromatographic run, or rather in a single chromatographic run. Okay, so we use cellulose as the cellulosic material as the stationary phase. Okay, so cellulose, you know, everybody knows that it is a polymer which is found in plants. Okay, it is a structural polysaccharide, you can say, and it is made up of glucose molecules. Okay, so monomer composition is just glucose, and these glucose are linked to each other by beta 1,4 glycosidic linkages, isn't it? Everybody knows about it. So just to show you this structure, this is a linear polymer I have shown here. And then these polymer strings, you can say these are, or fibers are held together by weak interactions such as hydrogen bonding. In the lower diagram here, you can see the hydrogen bonds. Okay. And by virtue of these hydrogen bond, fibers are held together, and the structure of the paper is formed. Okay. A plane surface is formed over which we can carry out the separation. Okay, so coming to the more detailed part of the interactions, okay, what actually happens is there are sequential separations which are happening at every place or every point on the paper as the solvent molecule moves on the paper. How does the solvent molecule move? The movement is by capillary action. Okay, did I write somewhere here capillary action? I'll just try to see. Yeah. Yeah, I have written it here. Capillary action is also called as the capillary effect, or it is also called as the wicking action. Wick हा शब्द मी तुम्हाला सांगितला होता का मागे इलेक्ट्रोफोरेसिस मध्ये? I think I have told you the meaning of the
हो
word.
सर सांगितलं. दिव्याची वात बद्दल बोललो.
Yes, yes. Okay. So this is the kind of action which actually allows the liquid to flow. Okay, and the liquid flows in the narrow spaces which are present in the fibers. Okay, and the movement of the liquid is not opposed by the forces like gravitational force or any other force. Okay, and this happens because there are intermolecular forces. Okay, that is adhesion and cohesion. These are the two important forces which are there between the liquid and the surrounding solid surface, that is the surface of the paper. Okay, now if the diameter of a particular region, okay, where the liquid is moving between the fibers, if that diameter, you can consider it as a small tube, okay, tubular structure. If the diameter or the space is very, very small, then there is also the action of what is called as the surface tension. Okay, and because of the combined effect of all these forces, the liquid moves along the solid surface. Okay, coming back to my earlier slide, and as the liquid moves, okay, it is the liquid is getting bound or bounded by certain regions in the cellulose, and these regions are called as the compartments. Okay, now within each compartment, there is going to be what is called as the separation reaction which is happening. Okay, and this separation actually is an equilibrium phenomenon or equilibrium based reaction, you can say. So what is the meaning of this equilibrium based reaction?
So the forward reaction will be equal to the backward reaction.
Wonderful answer. Okay, so this is a reaction where, for example, I have a small part of the liquid, okay, solvent, which is carrying the solute. And suppose I have, for example, 100 molecules of the solute. Okay, now out of this, can I share another screen? I'll just try to share a whiteboard. Okay, if I require to use it, then I'll use it. Okay, so suppose I have this particular surface, okay, and I have applied the sample at this particular point, okay, and then the movement of the solvent is like this, okay. Now what happens is in this particular sample, suppose I have got 100 molecules. I cannot write very properly with the mouse. Okay, 100 molecules of the sample. Okay, then out of the 100 molecules, suppose I tell you that the equilibrium is such that 40 molecules are going to remain, okay, they are going to remain in the stationary phase because there is an interaction of the molecules with the stationary phase, and the remaining 60 molecules, okay, 60 molecules are going to move ahead with the mobile phase. Why? Because the 60 molecules are getting solubilized in the mobile phase. Okay, so the equilibrium reaction is such that 40% are sticking to the surface and 60% are moving ahead. Okay, now when they reach a particular point, let me show it in probably a different color. Okay, now when they reach at this particular point here, then once again this interaction was happening here, okay, at one point. When the mobile phase moves to the next point, okay, or next compartment, you can say, okay, again out of the 60 molecules, 40% are getting bound to the, okay, 40% of the 60 molecules are getting bound to the stationary phase, and 60% of what? Of not the 100%, 60% of the 60% which were remaining in the mobile phase, they will continue to move ahead. Okay, so they will continue to move ahead. So how much is 60% of 60? Any guess? My mathematics is very
30,
poor.
36, I think.
36. I'll take your answer as the right answer, okay, because I don't know mathematics. Okay, so maybe let me write that number also. So what I'll do is I will erase this number here, and instead of that I will write where is the pen कुठे असतो याच्यामध्ये? Okay, how much did you say? 36, is it right?
Yes, sir.
So out of the 60 molecules which were there in the mobile phase, when the mobile phase moves to the next point, 36 molecules from that 60 are going to go again with the mobile phase, and the remaining, I think, how much? 24 molecules, is it? 24 molecules, okay, they will remain bound to the stationary phase, or they will be retarded, in other words. Okay, when I say they will remain bound, it means that they will be retarded by the stationary phase. So again, when the 36 molecules are moving ahead with the mobile phase, when they go to the next compartment, okay, now the concept of compartment here is a theoretical one. Okay, you have to just imagine it. Okay, I cannot define you how much is the compartment exactly, how big is the compartment. Is it right? So it depends upon the interaction, how many points are there available on the surface for the interaction to take place. So when the mobile phase goes to the next compartment, out of the 36 molecules, again 60% of them are going to move ahead, and the remaining 40% are going to stay on, or they will be retarded by the stationary phase. Do you follow what I'm saying? So that is what I mean when I say equilibrium reaction, right? Okay, so I'll once again come back to my presentation, right? Okay, so the next sentence here is like this, that a test solutes or the sample components which are present in our mixture, they will get dissolved in the mobile phase, okay, and then they will encounter the different compartments as the liquid moves ahead, and within the compartment the partitioning is taking place. Now this partitioning is dependent upon the differential solubility, or you can say relative solubility of the sample components. So either they are more soluble in the stationary phase, or they are more soluble in the mobile phase. So accordingly, they will get distributed. Okay, and this distribution is essentially an equilibrium based reaction. Okay, now what happens is the mobile phase is continuing. Okay, fresh mobile phase is coming. So, when the fresh mobile phase come to that particular point where, can I come back to the, okay, that board is already gone. Why? Okay, it's still there. Okay, now what I am telling you here is this 40% of the solute molecules are still there on the stationary phase. Now, when the new mobile phase comes, okay, shall I show it with a different color? I'll try to. Okay, so when the new mobile phase comes here, this new mobile phase is again going to push the 40% of the solute molecules which are there on the stationary phase. Okay, and then what will happen? Again, the equilibrium will take place. So out of the 40% or out of the 40 molecules, 60% of the molecules are going to move ahead, and out of the 40% will remain back. They will be retarded. Okay, again this 60% are going ahead, 60% of 40% they are going ahead. Out of that, 60% again will move forward, and 40% of that will again remain retarded. They will be retarded by the stationary phase. Now you can imagine that this kind of interactions are taking place at each and every point during the movement of the mobile phase. So new mobile phase is coming, it is taking away 60%, okay, and 40% is remaining. From the remaining 40%, again the new mobile phase is coming. Out of the 40%, again 60% will move ahead, and 40% will remain retarded on the stationary phase. Okay, so this is happening continuously. This particular sequence of events, you can say, is happening continuously during the run of the chromatography. Right? Now, I was telling you about only one particular solute molecules. Now there is a mixture of solute molecules. Now the same kind of interaction is happening for the other molecules, but the equilibrium is a different equilibrium. Okay, so for another molecule, for example, if it is a B molecule, earlier one was A molecule, for example, the B molecule is getting partitioned in such a way that 20% will be retained and 80% is moving ahead. 20% retained, 80% moving ahead. Okay, so you can imagine that for this kind of molecule, which is more soluble in the mobile phase, it will keep on moving at a faster rate compared to the A molecule, right? And if I have another molecule, say C, and for the C molecule, the equilibrium is like this, that 50% of the molecules are retained in the stationary phase and 50% are moving ahead, isn't it? So now what can we say about the sequence of the events? The one which is getting 80% solubilized in the mobile phase, that will move very fast. Okay, then the 50% molecule, that is the C molecule, will be an intermediate molecule, and the A molecule, which is 40% getting retained on the stationary phase, that will probably be the, you can say, I said something wrong. The C molecule, which is getting 50% dissolved in the mobile phase and 50% in the stationary phase, that will be the least mobile. Okay, and the 40% soluble molecule, which is getting 40% which is getting retained on the stationary phase, that molecule will be the least mobile molecule. So it will move the slowest on the chromatography. Is everybody getting what I'm saying? Okay, of course I didn't show all of these molecules on the whiteboard. That would have made the things more clear to you. Okay, Yogita, Pooja, Shubhangi, are you following what I'm saying?
Yes, sir.
Yes,
Yes, sir.
Yes, sir.
कळणार नसेल तर सांगा, मग मी व्हाईट बोर्ड वरती पुन्हा दाखवेल तुम्हाला. Okay, then another thing which I wanted to tell you is, in other words, we can say that the solute which is retained more, okay, solute that is distributed preferentially in the mobile phase. Okay, it is more soluble in the mobile phase, that will travel faster, and the solute which is not distributed preferentially in the mobile phase, or rather it is distributed more in the stationary phase, that particular solute will move very slow, and therefore there is a separation of the two different types of solute, or there may be a number of different solute molecules in the same mixture. So all the solute molecules will get separated depending upon their own equilibrium characteristics. Okay.
Yes.
Okay, then another thing which is important is the more the partitioning reactions happen, the more will be the separation efficiency. What is the meaning of this sentence? The sentence means that if you have a bad quality chromatographic paper, okay, a simple filter paper you can say, or blotting paper you can say, where the paper itself is not very uniform, okay, and if that is the case, then the partitioning reactions that will take place will be lesser in number. The compartments which are available sequentially, the compartments will be lesser in number, and therefore the partitioning reactions will be lesser, and therefore the efficiency of separation will be less. Okay, if the paper is of a good quality, very fine quality paper, then the compartments which are available for the partitioning reactions will be more. Okay, so more partitioning reactions will take place, and therefore the separation efficiency will be very good. Is it okay?
Yes.
Okay, so coming to the partition coefficient, I think I forgot to write this title here. I'll write it here, partition coefficient. Okay, so can I save this? Let me try. Okay, that is given by this formula. K is equal to Cs divided by Cm. Cs is the molar concentration of solute in the stationary phase, and Cm is the concentration of the solute in the mobile phase. Okay, now the rate of movement or rate of travel of the mobile phase, it will depend upon certain things. Okay, so one is the velocity of the mobile phase. Okay, how fast the mobile phase can travel from one end of the paper to the other end, and of course all the sample components will have this the same kind, will be under the effect of the same kind of velocity of the mobile phase. Okay, so separation will not happen because of this actually. Okay, the velocity of the mobile phase is only responsible for carrying the sample components along the paper. And the other important factor is the ratio of the volume of stationary phase to the mobile phase. Okay, so more is the amount of the stationary phase, probably the retention behavior will be more, isn't it? And therefore the rate of travel of the sample components will depend upon the ratio of the stationary phase to the mobile phase in terms of the volume or amount, you can say. Then the important factor again is the relative solubility or the partition coefficient of each solute in the mixture. Okay, and the fourth important characteristic is the polarity of the mobile phase. Okay, so more polar is the mobile phase, then the polar solvents will move faster. Okay, and if the mobile phase is less polar, then the less polar solutes will travel faster with the mobile phase, right? So here I have given you a series of the solvents which are arranged in the you can say increasing order of the polarity. Okay, so hexane, for example, is the least polar solvent, that is followed by cyclohexane, benzene, chloroform, and so on and so forth. Then we have methanol, which is more polar than any of the others. And then distilled water is the most polar solvent which is used in chromatography. Okay, so water is the highly polar solvent and hexane is the least polar solvent. Then I have told you about the capillary action, that this is a diagrammatic representation showing you how the separation is taking place. So we apply the sample towards one end of the paper, okay, and then this paper is dipped in the solvent system, okay, in such a way that the spot actually is not in contact with the liquid. Okay, otherwise it will just get dissolved in that solvent system. Okay, and then by capillary action the solvent will move up along the paper. Okay, so this is a typical you can say example of what is called as an ascending chromatography. Okay, and as the solvent,
Descending.
yes, any
No,
question?
no, sir. No, no.
Okay. Okay, so as the liquid moves, it will carry the sample components along with it. And then depending upon the equilibrium characteristics of each sample component, they will get separated from each other. And then you will realize here that as the solvent moves ahead towards the other end, the separation is getting better and better. Okay, separation is getting better. In other words, can I tell you here that the longer is the paper, okay, longer is the strip of paper, the better is the separation. Is it correct? If I take a very short piece, small piece of paper, then probably the separation will not be very clear. Okay, if I take a very long piece of paper, then the separation will be more. But always there is a limit to how big a paper you can use, because the chromatographic paper is a costly paper. It is a good quality paper, isn't it? High quality paper. It is costly, isn't it? So it always depends upon your affordability. Okay, how much, how big a paper you can use, and which will be able to give you proper separation in your experiment. Is it right? Okay. Then we go to the
Yes,
next
yes,
slide.
sir.
And this slide tells you about another term which is called as the retention factor. So retention factor actually is a degree of retention of a particular solid molecule. Okay. For example, in this sample, we have 1 and 2. Two different components are present in the mixture. Okay. The spot was placed here. Chromatographic run was allowed to carry. And then at the end of chromatography, I have got these two spots, a blue spot and a red spot. And then what I'm going to do is using a scale, I'm going to measure the distance which is traveled by one particular solvent from the point of origin. Okay, point of origin here. Okay, from this to this distance. And then for the other spot, I would calculate the distance from this point origin to the center of the spot. Okay, center of the spot, this word is very important. Okay, and then I also calculate the distance which is traveled by the solvent front, this line here, that is the solvent front. Okay, and then for each particular component I calculate the RF value like this. RF value is the distance which is traveled by the molecule, that is this distance. For example, here it is 0.4 centimeter, you can say, or 0.4 inch, whatever you want to say, divided by distance traveled by the solvent. So from this point here to this place here, and that is, for example, 1 unit they have written here, maybe 1 inch or 1 centimeter. Okay, and therefore the RF value for this component is 0.4 divided by 1, that is 0.4. And for the red spot here, the RF value is 0.8 units divided by 1 unit, that is 0.8. Okay. So if the retention factor is more, RF value is more, then what does it mean? It means that the distance traveled by that particular component is more. Okay, 0.8, right? And then you will also be able to understand that the value of retention factor is never more than 1. Okay, because it is a ratio of the distance traveled by the molecule to the distance traveled by the solvent front. The distance traveled by the solvent front is always more than the distance traveled by each component. Okay? So your denominator value is always, कोणाच्या माईक मध्ये आवाज येतोय, त्यांनी बंद करा माईक. Okay, distance traveled by the solvent front is always more, and therefore the denominator value is always more compared to the numerator value, and therefore the RF value can never be more than 1. It is always 1 or less than 1. Okay, But if the RF value is 1, is it a good separation? It is not a good separation because your sample component is moving with the mobile front. It is not getting retained on the stationary phase. Okay, and therefore the RF value is 1. Okay, you should always adjust your solvent system in such a way that the RF value is less than 1 so that you get the spot on the paper. You can visualize. How do you visualize the spots? Any idea? Have you done any chromatography, paper chromatography, and seen the spots?
Yes,
Yes, sir.
sir.
Okay, I think I should have added one slide here. That is detection. For example, I will just write here that if you have amino acids, then they are detected by a reagent called ninhydrine. Okay, I think we
Anhydride
have
solution.
already done this in some time. Okay, we are also going to perform a practical here. Okay, and in that practical, we are going to carry out the separation of amino acids using paper chromatography. Okay, we are also going
Yes,
to do thin layer chromatography. Of course, if we get time for all that. Then we go to the applications of paper chromatography. And then you can see here that there is a large variety of different applications of paper chromatography. One is to check the purity of pharmaceutical compounds, okay, or drug molecules. We can carry out the separation of the fermentation products, okay, and therefore we can study the process of fermentation, whether the fermentation is all right or whether it is complete or whether it is incomplete. All that we can find out. We can inspect the cosmetics, different types of cosmetics, whether they are of a good quality or not. We can detect the adulterants in the food. Okay, food adulterants माहिती का तुम्हाला? What are adulterants?
Yes, sir.
yes, sir.
Example
FDA.
of- Yes. What did you say? Shamim,
Yes, sir.
did you say something? Okay, so adulterants are something, some you can say impurities which are added to the food to increase the bulk quantity of the food. Okay, of course that is not a good practice, you know. Then we can detect contaminants in drinks and foods. We can examine some biochemical reactions or biochemical investigations can be carried out using paper chromatography. And then it is also used for testing the dopes and drugs in humans and animals. You know what is dope? Doping करणं माहिती आहे ना?
Dopamine.
नहीं, not exactly dopamine, but something which is taken by the
ते
sportsmen, sports people.
ऑलिम्पिक मध्ये डोपिंगला ते करतात सर बाद
Steroid,
करतात
steroid घेतात ना?
हा स्टेरॉइड सर
Yes, that is called doping. Okay.
Illegally घेतल्या जातात.
Yes, yes, illegal things. Okay. And then we will also go to types of paper chromatography. I'm not going to spend a lot of time. I expect you all to read about it from different sources, which I have given you already. Okay, so there is something called as an ascending paper chromatography, where the movement of the samples is from the base of the paper. Okay, it's against the gravity, we can say. Okay, from bottom to the top. And then we have descending chromatography, where the flow of the of the mobile phase is from top to the bottom. Okay, top to bottom vertically. That is called descending paper chromatography. Now what in the case of descending paper chromatography, there are two different forces which are used to drive the solvent. Okay, one is the gravitational force and the other is the capillary action. Okay. So now between the two types of chromatographies, ascending and descending, which one is better and which is not better? Can you say something? In descending paper chromatography, I can tell you that because of the two forces which are applied, the speed of separation is more. Okay. So you can get faster separation in descending paper chromatography. Whereas in the case of ascending paper chromatography, because it is going against the gravity, probably the rate of movement is slower, and therefore the chromatography will take longer time. Okay. But on the other hand, descending paper chromatography, because the rate of movement is faster, you can say that probably the separation is not very good. Okay, separation effect is not very good. Whereas in the case of ascending paper chromatography, the separation can be better. Okay, because it is taking a longer time, but the separation will be better. Okay, but usually what we do is, if you want to get better separation in descending paper chromatography, what one can do is one can take a longer piece of paper, okay, longer strip of paper, and therefore the separation will be better, because I have already told you that longer the piece of paper, the better will be the separation, isn't it? Then there is another type of chromatography called as radial chromatography, radial or circular, where you apply the sample, you take a circular piece of paper, okay, and you apply the sample at the center, and it will spread on in all the directions, and it will form a circle. Okay, so instead of getting a spot on the paper, you will get a circle on the paper. Is it right? Can you say anything about what could be the advantage of this technique?
Separation of mixture.
Yes.
Separation of a mixture.
नाही ऐकू येते मला. Can you repeat again?
Separation of a mixture.
Quality
Organic
and
inorganic
quantity.
solvent.
सेपरेशन आपण करतोच आहे ना याच्यामध्ये. Compared to ascending and descending paper chromatography, what is the advantage of radial chromatography? That is what I'm asking.
So better visualization.
Yeah, maybe. Yes. Okay. Visualization will be more better. The sharpness will be more, because instead of getting a round spot, sometimes the spot may not be exactly spherical. Okay, the
So
shape,
maybe we can get a whole zone. I
you can
think we will be getting a zone.
getting a zone. Okay,
Yes.
you get a sharp line, more sharp line in case of radial chromatography. Okay, and therefore for calculation purpose it is better. You can exactly measure the distance traveled by the sample component from the center of that circle. Is it right? You can, if the
Yes.
shape is not exactly circular, still you can find the distance migrated from the center to different points on the circle. Okay, and that will help you to calculate the average more correctly. Okay, average values can be obtained more correctly, right?
A color spot different space
Yes,
in
yes,
this technique.
yes. I didn't get.
Descending chromatography. Uh, spot
Yes,
connectivity problem.
कनेक्टिविटी प्रॉब्लम है.
Color
बंद भी होने वाला है 4-5 मिनट में, जल्दी बोलो.
color spot different position observed in this technique. Descending chromatography.
आई डिडंट गेट यू.
Okay.
चैट करोगे क्या? You can chat and then I will see what you want to write.
Okay.
Okay, and then lastly
Okay.
come to a two-dimensional paper chromatography. Just as we have seen in the case of two-dimensional electrophoresis, again, if the two different components are having properties very similar to each other, or polarities which are very close to each other, then by using two-dimensional chromatography, we can easily separate the molecules. Okay, we can use a different solvent system, for example. Okay, इथे लिहायचं माझं राहिलेलं आहे. Two different solvent systems can be used one after the other. Okay, so what we do is we put the paper in the solvent system, allow the chromatographic separation to take place in one dimension. After the running over, we remove the paper, okay, and then we change the direction of the paper 90 degree, then again put the paper in another solvent system. So if you use another solvent system, it means that the polarity is changed, and because of the different polarity of the mobile phase, the components which were close together forming a single spot, the two components will get easily separated from each other. Are you getting what I'm saying? In case of two-dimensional electrophoresis, we had used two different techniques. For example, if one was tube gel electrophoresis or isoelectric focusing, okay, the other was SDS phase electrophoresis in the second dimension. So similarly here we use two different types of solvent systems so that we can separate the sample components which are having similar polarities in one solvent system. Okay, similar polarities in one solvent system and different polarity in another solvent system. Is it right? Okay, detection part we have already covered. And in the next lecture, I will try to cover thin layer chromatography. Okay, but I just want to remind you one thing, that is on Monday and Tuesday, I'm going out of Aurangabad for an official duty. Okay, I'm not available on Monday and Tuesday. So Tikekar sir will continue. Okay, and if there is some other teacher, maybe that teacher also will engage your lectures on Monday and Tuesday. Okay, if you have any questions, then you can ask me. I have only, I think, 3 to 4 minutes are available.
So what exact RF value indicate?
RF value indicates the degree of the retardation of one sample component. Okay, degree of retardation on the stationary phase. Okay, it is retention. Retention means the degree of retardation on the surface. And then we can compare two different sample components. Okay, the one which has a higher retention factor is actually moving faster. Okay, as per the formula that we have used. Is it right?
We can also calculate the specific gravity, I think, with RF.
Specific what?
Gravity.
Gravity? No, no, no. Not gravity. What do you mean by that? Gravity? Tikekar sir is ready? No, no. In maybe another 5 minutes he will engage your lectures.